So we are moving on to our second primary meristem, the ground meristem, and looking at some of the tissues, the primary tissues that come out of that. So this could be in a root or a shoot. If it's in the root, we would have gotten our ground meristem from the root apical meristem, the ram. If we're in the shoot, then that ground meristem would have come from the shoot apical meristem. But these sets, the apical meristem and these first three primary meristems, will always be the same. So ground tissues. Ground tissues can be simple. They can either be fully parenchyma, they can be fully collenchyma, or fully sclerenchyma, or they can be complex, where they're made up of some mixture of those cell types. Here is an example of some ground meristem products. So the ground meristem would divide to produce these primary tissues. So the primary tissues you're looking at here they're separated into two distinct regions, so we're going to give them special names. This outer region that's between the epidermis and the vascular tissue is going to be called the cortex. So it's fully defined, that term, cortex, because there's this region where it exists, where it's between the epidermis and the vascular system. Here is another example of what that can look like. As a plant gets older, that cortex often gets smaller and smaller, but there's still this thin region of tissue between the epidermis and the vascular system. So we still have some cortex out here. And in this cortex, we have some collenchyma, some bendy support cells, and then we have a bunch of parenchyma. So the cortex is just this thin, squished region of cells out here that's composed of collenchyma and parenchyma. The tissue on the inside of the vascular tissue, so it's fully surrounded by vascular tissue, is called the pith. So you can see that here in this like brighter peachy color, and here it's just blue still, but that's just a difference in the staining. So the pith is completely surrounded by vascular tissue. So that is what it looks like usually in eudicot stems, where we'll have this distinct cortex and pith. In eudicot roots, we just have a cortex. And then in monocot stems, we end up having this arrangement of vascular tissue where they're arranged in bundles that are arranged in concentric circles. And they don't make a distinct ring each time. They're sort of located in a sort of ring, but they don't form an entire ring or a cylinder. So in this sense, there's no real region that's between the vascular tissue and the epidermis or fully surrounded in vascular tissue. So we just call this stuff surrounding the vascular tissue ground tissue. So depending on the arrangement and location of vascular tissue, our tissue produced by the ground meristem will have a different name. If it's distinctly between the vascular tissue and the epidermis, it's called the cortex. If it's distinctly surrounded by vascular tissue, we'll call it pith. If there is no organization or distinct regions, we'll just call it ground tissue. So many times, most often with plants, things are named and identified based on their location. So that is part of a theme that will run throughout this whole segment on anatomy and physiology. Okay, so that's the ground meristem. The procambium is going to make the primary tissues of xylem and phloem. So primary xylem and primary phloem. And this one doesn't have as intuitive of a name, procambium, but it'll make sense later once we learn about some secondary tissues. So I'll explain it later. So the vascular tissue system is made by the procambium. And the vascular tissue is complex. Of all of the tissue types, it contains the most different types of cells. So first we'll look at the xylem. And I have this as sclerenchyma, but it's not. So xylem is made of specialized cells that look like sclerenchyma, but they're not sclerenchyma because they're not produced by the ground meristem. They're produced by the procambium. So xylem transports water throughout the plant. I'll just keep deleting this as we go along. You can watch me edit my slides in real time. So within the xylem, we have a few specialized cells. So in your lab, I talk about general cell types. That would refer to parenchyma, collenchyma, and sclerenchyma. I'll also talk about specialized cells. So within the dermal system, we saw guard cells and trichomes as these examples of specialized cells in the epidermis. They were still parenchyma cells, but they had specialized for a particular function. So here, within our vascular tissue, we're going to have a lot of specialized cells that aren't necessarily going to be a certain cell type. So tracheids are one of our specialized cells within the xylem. They have thick secondary walls, and they are dead at functional maturity. So they're very similar to sclerenchyma in their definition, but they're not produced by the ground meristem. They're long, narrow cells. There's an image of them here. And they have these bordered pits in these sets all up and down the sides. And these are going to transport water in a weird way. So instead of just transporting water like a pipe, they're going to transport water through these bordered pits in pit pairs that will line up with an adjacent tracheid. These are the earliest of our lignified vascular tissue. So we would see these in seed-free vascular plants and gymnosperms. And this would be the only type of cell in their xylem. In angiosperms, we get a new type of xylem specialized cell. We get vessel elements. We also see these in the neophytes, which is really weird because they're within the conifers, so they independently evolved these vessel elements. Similar to tracheids in their function, but completely different in the way they are shaped and how they transport water. So tracheids have all these bordered pits where they have to transport water from side to side through very small holes. Vessels are still going to have those bordered pits, but their main way of water transport is going to be straight up and down through these perforation pits that are located at the ends. It's just a big hole that's lined up with the cell above and below it. So it really does just form a pipe. There can still be water transferred between those bordered pits, but the main water transference just occurs through these perforation plates at the ends. They're super wide. But again, with anything, there's no good like size definition for this. Vessel elements can be just as narrow as tracheids or far wider. But when you see a really wide cell, you know it's a vessel element. And tracheids won't have these large perforation plates at the end. So here's an example of the sort of variation between vessel element sizes. Here we have one that is really short and wide, but all three of these are vessel elements, right? They can be extremely narrow, and here we have tracheids, which are, just as narrow as those ones. And then this one here on the far right is a fiber. This one actually might be a vessel element too. Now I can't tell because it kind of looks like it has the weird wall thickenings. So difficult to tell apart unless they're super wide, but we have some overlap in size between tracheids and vessel elements. But tracheids will not have that perforation plate up at the top. So the other part of our vascular system, we have xylem to transport water, and that goes in one direction from the roots up to the shoots and out the leaves. But we also need to take our sugars that are made in our above ground parts and be able to transport them to the inside of the plant as well as to the below ground parts that aren't able to do photosynthesis because there's no light. So phloem tissue needs to be able to transport in different directions, not just down, right? It needs to go all around the plant. So phloem tissue is going to be alive, unlike xylem tissue, which just goes in one direction and is regulated by these sort of cohesion, tension, suction of water. So phloem cells are alive. They're parenchyma cells, and they transport photosynthates, which is a fancy name for the sugars made from photosynthesis. The phloem tissue is made up of different types of cells. So in early plants, like seed-free vascular plants and gymnosperms, the phloem is only going to contain, for the transporting cells, the conducting cells, sieve cells. So sieve cells are long, narrow cells and have tapered ends with regions called sieve areas. So really analogous to a tracheid, but they're alive and they're transporting different materials. So here, oh, maybe I don't have a good picture of sieve cells in here. Dang it. So in angiosperms, again, we specialize, we get better, we come up with a better idea. So angiosperms have cells called sieve tube elements. You won't see these in gymnosperms. You'll only see them in our flowering plants. So sieve tube elements have a sieve plate instead of these sieve areas that have to try to line up with each other. Once again, we're making a solid pipe with a plate that's kind of like a grate. And it's rare if you're looking at a cross section that you'll actually get to see one of these plates because they're only, spaced every so often at the ends of a cell. So you have this sieve plate, and these cells are empty. They don't have contents in there. They have still cytoplasm, but they don't have like a nucleus and organelles, but they're alive. So to function, they have this smaller companion cell next to them that has a nucleus and controls all of the function of both the companion cell and the sieve tube element. So this is this new weird specialized system. If you've ever watched Mad Max, the old ones, where they have Master Blaster, the little guy on the big guy's shoulders and the little guy's the brain, that's like the companion cell, and the big guy is the sieve tube element that does all the stuff but needs something to tell it what to do. So sieve tube elements are only in angiosperms, and they will be accompanied by those companion cells. And if we were to look at this in a cross section, here we have a sieve plate of a sieve tube, and we have this tiny little companion cell next to it. And this would be found in the phloem tissue, so within a vascular bundle or vascular cylinder. This clump of stuff here is called P protein, and you can think of that as phloem protein. It coats the sides of the sieve tube elements so that when a plant is damaged, then that P protein can rush down to a sieve plate and plug it. So it forms something called a slime plug, and it's kind of analogous to a scab, where it's to keep the valuable sugars from flowing out of the plant in case of damage. Here's an example of our phloem cells here. We have sieve cells, again, very similar to our tracheids, our sort of analogous cell in the xylem. And then we have sieve tube elements, which are analogous to vessel elements, but these are alive and they need a brain to help tell them what to do, and that's the companion cell. So sieve cells will be in seed-free vascular plants and gymnosperms. Sieve tube elements and companion cells will be found in angiosperms. The other cell here is a sclerenchyma cell, this long spindle-shaped fiber. So we get bundles of phloem fibers in or adjacent to our vascular bundle because xylem is lignified, so it forms like a solid pipe, but the phloem that has to travel next to it all around the plant is parenchyma, and so it's kind of like flimsy. So if you have this clump of phloem fibers next to it, then that provides this rigid support for your vascular bundle on either side. So helps provide structure and protection for the vascular bundle to travel around the plant. So that's our last component here. And I don't know if I think they're still technically sclerenchyma. I think they might be made by the ground tissue. But actually, I don't know. So fibers are these long tapered cells with a thick secondary wall and lumen. You can find these in the vascular tissue. You can also find them just within the cortex. They provide structural support. And this is what they look like. So if you have a cross-section and they're stained with that stain that gets the secondary wall red, they'll just look like these really thick-walled cells. Often they're in clumps. If you're looking at them, like you take some of your toilet paper and look at it under the microscope, you'll see these long, thin, dead cells. Those are fibers. That's what our paper and clothing and wood and things that we use wood pulp for, or woody plants, are those fibers. And here we see the vascular tissue organized. So it can be organized into vascular bundles, where they make like a distinct vein, or a vascular cylinder, where it makes a big tube of vascular tissue. So here we'd have the epidermis, the cortex, and this vascular cylinder. There might be a tiny bit of pith in here. So here, big vascular cylinder. Here, as we'll see in our celery cross sections, we have a bunch of vascular bundles in these distinct bundles, with the xylem always toward the inside of the plant. Okay, so a little review here. We have our apical meristems that make our primary meristems. We can have that in the shoot and the root, and there'll be many. The plant starts out with just one, but then each of those will then make new branching parts, and each branching part has its own apical meristem. Our vascular tissue can be formed in cylinders or bundles, and within the vascular tissue we can have xylem, which has thick secondary walls and is dead at functional maturity. It's always oriented toward the inside of the plant. So this is our xylem here with our huge vessel elements and potentially some tracheids surrounding them. These will be transporting water. Toward the outside of that vascular bundle, or outside of the plant, so this is closer to the epidermis, we would have our phloem tissue, and that's composed of these larger sieve tube elements and our smaller companion cells. This is an angiosperm. We'd be looking at corn. And here is a bundle of phloem fibers. So these are helping provide structural support to this vascular bundle. If we were to look at these cells from the side, they can have crazy secondary wall thickening patterns. You're likely to see these in some of your samples, and it'll look like a spring traveling through. So if you see them from the side, they look really cool. Just wanted you to see what they look like. And this is what the bordered pits look like they have a little donut surrounding the hole. Alright, so there's some with some labels. You can pause on this slide if you'd like to. And then I will save the rest of these slides for our roots and shoots lectures. Okay, I think you have all you need for this week's lab.